Hey all, welcome back. In the past, we've talked about how gene expression can be controlled at the level of transcription, by DNA modification, by RNA processing, and also by the stability of RNA and protein. In this video, we're going to talk about more recently discovered mechanisms of gene expression. We're going to talk briefly about how microRNAs and small interfering RNAs are involved in a process known as RNA interference, and we'll discuss how CRISPR in bacteria is used to control the expression of phage genomes. What I'm showing you here is the structure of a basic mRNA after it has undergone RNA processing. Let's take a minute to review this structure before we talk about how microRNAs and siRNAs control the expression of proteins that are generated by this mRNA. You may recall that at the 5' most and the 3' most ends of this mRNA, we would have a cap and a poly A tail. Recall that the cap and tail are not only involved in the stability of the RNA, but they are also important for the translation of this mRNA. Between the AUG, which represents our start codon, and the UGA, which represents our stop codon, we have what's known as the coding sequence. This is the region of the mRNA that codes for a protein. Between the 5' cap and the AUG, we have what we call the 5' untranslated region, or the 5' UTR, and we can sometimes find regulatory elements within this region. The focus for microRNAs and siRNAs are going to be this region here, which is known as the 3' untranslated region, or 3' UTR, which occurs between the stop codon and the poly A tail. Now in this image, what we're looking at is a microRNA and an mRNA, particularly the 3' UTR of an mRNA. MicroRNAs and siRNAs are typically 22 nucleotides in size. I'm going to be using microRNA and siRNA synonymously today, but I want you to note that microRNAs are naturally occurring phenomena within cells. siRNAs are typically made within a lab and then injected into organisms to artificially repress gene expression. So let's get back to the structure of a microRNA here. As I mentioned, microRNAs are going to bind to the 3' UTR of an mRNA. As you can see in this example here, we have a short region of about 7 or 8 nucleotides that are perfectly complementary between the microRNA and the mRNA. This region where this perfect complementarity occurs in the microRNA is known as the seed sequence. And this seems to be the most important sequence for microRNA to inhibit the expression of mRNAs. There may be some binding as well at the 3' end of the microRNA, but this binding is much less frequent. I also want to point out here, you may notice those 2 dots between the G and the U toward the end. GU base pairs can form, but they are incredibly weak in comparison to AU and GC base pairs. And so we do see some weak association between G and U in the case of a microRNA with an mRNA. Now, microRNAs are initially synthesized in the same way that all RNAs are synthesized, and that is by transcription through RNA polymerase. Initially, a primary microRNA is produced. In this image here, what is going to become the mature microRNA is shown in red. This primary microRNA is cut into a hairpin loop structure known as the pre-microRNA. This is exported from the nucleus and then cut again by an enzyme known as dicer into a microRNA duplex. One of the 2 strands in this microRNA duplex will become the mature microRNA, and it will associate with an enzyme complex known as RISC, or RNA-induced silencing complex. RISC can control gene expression in a number of ways. In the case of endogenous microRNAs, what most often occurs is that RISC inhibits the association of translation initiation factors with the 5' cap of an mRNA. However, RISC can induce the degradation of the mRNA in cases where the microRNA, or more likely the siRNA, is base pairing perfectly with the 3' untranslated region of the mRNA. In these cases, RISC can induce deadenylation of the mRNA. In other words, it can remove the poly A tail. It may recruit enzymes to decap the mRNA from the 5' end, or it may induce what we call slicer activity, where the RISC complex cuts the mRNA right in the middle of the 3' UTR. All three of these events can lead to a quicker degradation of the mRNA. So in essence, the takeaway point here is that microRNAs repress translation or increase the rate of mRNA degradation, both which will cause a silencing or a repression of gene expression. Now, in the past few years, there's been a lot of attention paid to a new form of regulating gene expression, and that mechanism is known as CRISPR. CRISPR has been proposed to be used as a gene editing tool in eukaryotic cells. However, the basic mechanism of CRISPR has been worked out in bacterial cells, where it was originally created. In CRISPR, what we find is an immune system of sorts for bacterial cells. Bacteria, like human cells and other eukaryotes, are infected by viruses. And in the case of bacteria, what we find is that when the virus injects its DNA into the cell, this DNA can be taken into regions that are known as CRISPRs. These CRISPR regions can be transcribed. They will associate with molecular machinery known as the Cas system. And this Cas system, along with the CRISPR RNA, if it ever sees the same virus again, can cause cleavage of that viral DNA. So this is a protective mechanism that bacteria are using to prevent infection, but in essence it is another mechanism of gene expression. It's likely that in the near future we'll see a lot of work invested in CRISPR, determining whether we can use it to control gene expression in human cells.